Hi everyone, my name is Sarah. Today we're going to talk about the Leucine zipper. And this is the Group Advanced Concepts presentation number 1. Okay, so a big part of the Leucine zipper is the coiled coil, which you can see on the right. There are the two twisting structures that wind around each other. These are two alpha helices that are forming a supercoil by that winding action around each other. And it's actually the two side chains that are interacting. And the side chains in this case are lysine and glutamate. The lysine residues occur around every 7 intervals. Kind of how it's working is the positively charged side chain interacts with a negatively charged side chain. So there's this electrostatic reaction, or opposites attract reaction, that is occurring. As we could see from that last picture, you saw the two big blue winding structures that form that coil. Well, both of those were alpha helices. So what is the alpha helix? You can see on the first picture on the left with the R groups, or the side chains. So there's a chain of hydrogen bonding that is going to occur. And if you look at the picture on the right, you can see the CO and the NH. The hydrogen bonding that occurs is between the CO and those NH groups. And you can see that they're kind of running along that main chain there. A hydrophobic reaction is what increases or keeps that strong core, it keeps that stability of that complex itself when the hydrophobic amino acids are protected from that aqueous media. Okay, in our prompt we wanted to know one possible residue that could replace leucine, and for this prompt I chose isoleucine. Isoleucine is a great fit because it can maintain that stability that is essential for that alpha helices coiled structure. So we know it can keep that nice and strong. We know that we need that, so that would be a great choice. And isoleucine does this by being equally hydrophobic. It has similar characteristics, such as the same chain length. And I chose serine for a destabilizer. And I chose serine because it's hydrophilic by nature. We know that we would need something that is hydrophilic. That's why serine would be a great choice. We know there's that polar core, and it's not going to be able to create that same reaction. As you can see here, there's the leucine and isoleucine in that upper right-hand corner. And if you look towards the bottom of both of those, you can see all of those similar C atoms. That's showing us another characteristic that they're similar and could propose that an isoleucine could propose that similar reaction that we need. Okay, for prompt 2, propose a different pair. We need a different amino acid pair whose side chains can interact in a similar manner to lysine and to glutamate. We had talked about both of these earlier and how one of them has a positively charged R group or side chain, while one of them has a negatively charged R group or side chain. And for this to be possible, we know we require a pair that contains one acidic and one basic amino acid. And having one acidic and one basic amino acid is what will essentially allow for that interaction that is similar to that leucine and that glutamate. For this, I chose aspartate and histidine. All right, the picture on the left has the hydrophilic and hydrophobic running along that left side. And we know that we need hydrophilic amino acids for this. So we can see the aspartic acid there, we can see that histidine, and if we go over to the right side of the slide, we can see along the top there are the positively charged R groups there, and then on the bottom we have the negatively charged R groups, or side chains. So as we had talked earlier about the lysine and the glutamates, one of them has a positive charge, while the other has a negative charge. So in order to have something similar, we need one of each, which is why I proposed histidine, which is one of the positively charged R groups, and the aspartate, which you can see is one of the negatively charged R groups. And that is what I chose for that. And that is all. So thank you guys.